Hello everyone, welcome back to Basic and Applied Microbiology classes. I hope you all are doing good. So today we are going to talk about mechanism of drug resistance. So before moving ahead and talking in detail about this topic, let's take introduction first regarding drug resistance, what actually it is. So drug resistance, we can define it like this. It is simply can be defined as reduction in effectiveness of a medication in treating a disease. Okay, so what actually happen in case of drug resistance? Suppose we have formulated a drug, okay, and this drug is designed specially to target a particular disease, and now drug is not responding to cure from that disease, to treat that particular disease for which actually it was designed. It means this is an issue of drug resistance. Okay, now if we say in microbiology terms how we can define drug resistance, so drug resistance actually results when microorganisms develop the ability to neutralize the effect of a drug which was actually designed to kill that microorganism. Okay, so how we can say in microbiology that microorganisms, suppose there is a pathogenic bacteria, say I am going to talk about Mycobacterium tuberculosis. Okay, we all are aware about it that it is known to cause tuberculosis disease. So we have designed a drug to treat tuberculosis. Okay, initially Mycobacterium was responding well and we were getting good results while we were using this drug to treat tuberculosis. But later on, what happened? Microorganisms, Mycobacterium has developed some special characteristics. Okay, it has acquired some kind of characteristic features, some properties that is allowing Mycobacterium now to resist the presence of that drug. Okay, and we are not getting any result from that drug. We are not able to get any cure from that drug against tuberculosis. Okay, so it means what? It means microorganisms, when they become insensitive, they don't show any sensitivity towards a drug which we have specially designed to get cure from the disease those microorganisms are causing. It means this is the case of drug resistance, and it is called as microbial drug resistance. Okay, now question comes, what are the sources, what are the causes, or I can say what is the reason why drug resistance actually come into existence, why drug resistance cases are becoming so common? So you should know that there are several causes of drug resistance. First, extensive drug treatment is one of the major issue. Okay, drug we are going to use to cure a disease. If of course extensively we are going to use such kind of treatment, we are going to use drug based treatment. It allows it provides the microorganisms platform to develop such kind of abilities, to develop such kind of characters that help them to resist the presence of drug. Secondly, if I say failure to complete the course of antibiotics. Okay, so first extensive drug treatment, it is followed by another issue. Another issue is what? Failure to complete the course of antibiotics. Suppose we are visiting a physician and physician is going to recommend us some antibiotic course that we need to follow this course up to 6 months. And what we are finding, patients generally find that after 3 months we are recovered from a particular disease and we stop the intake of antibiotics. So this time duration, this incompletion of recommended courses of antibiotic by the physicians allow the microorganisms to take the advantage of becoming resistant towards that drug. Okay, so now third issue is what? Use of suboptimal drug antibiotic dosage. If we are not using the right amount, right type of dose, we are going to take higher amount, higher dosage of antibiotic, we are going to take low dose of antibiotic. These are also creating some opportunities to for microorganisms to develop resistance against the drugs. Okay, the non-medical usage, I think many of you will not be aware about this, that antibiotics are nowadays also are in use in food industries for food preservation. Various kind of meat, fish, they are actually preserved by antibiotics. So this is actually misuse or indiscriminate use of antibiotics, which is also allowing microorganisms to take the advantage of becoming resistant towards different kind of drugs. Okay, so now you see unregulated sale of antibiotics, this is the another issue. So all these are actually the causes, the reasons which are providing microorganisms the platforms to avail the opportunity to become resistant towards different drugs. Okay, so now you see, one thing I would like to tell you, that the emergence of drug resistance issue has become, or it has been reported as a serious threat to public health in 21st century. Why? Because you see, we are pharmacologists, researchers, scientific communities, they are working day and night to develop drugs to target diseases. Okay, and what is happening, if microbial disease, microorganisms which are known to cause a particular disease, they are becoming resistant towards a particular drug, then what is happening, of course, we need to look for some new drugs. And during that time, what is happening, the patients which are suffering from that particular microbial disease, they will not get recovery on time, they will not get cure from that particular disease, of course. Then it is going to present what serious threat to public health. And one report is there from United Nations 2016 report. What it states, it states annually a death of around 7 lakh people results from drug resistant infections. And they have also speculated in their report, they have also reported this, stated this point, that this data is supposed to reach 10 million by 2050. So through this data, we can better understand that how important issue it is of drug resistance. Okay, so now question comes, what kind of bacteria are there which are becoming drug resistant. Okay, so you should update your knowledge that initially the cases were reported for gram positive bacteria. Okay, in initial cases, gram positive bacteria were there which were showing resistant towards drug. Okay, and nowadays various gram negative bacteria, especially those gram positive and gram negative bacteria which are found in hospital environment, which are known to cause nosocomial infections, also hospital borne infections, they are rapidly developing and transmitting antimicrobial resistance. Okay, so here I have also given name of a few: methicillin resistant staphylococcus aureus, MRSA, maybe many of you have heard about it; VRE, vancomycin resistant enterococci; Acinetobacter baumannii, Pseudomonas aeruginosa, and members of family Enterobacteriaceae, including Klebsiella pneumoniae, Escherichia coli, Proteus species, they all are showing resistant towards different drugs. Okay. Now question comes, what are how these bacteria develop the ability, okay, how they are having such kind of properties that they can easily develop resistance and they can transmit antimicrobial resistance. Then you should know that, of course, the principle is what, genetic basis. Okay, they are actually having genes for drug resistance. These bacteria, they are possessing genes for drug resistance, and where these genes are present, they are present on genetic material. Okay, genetic material means bacterial chromosome is there, extra chromosomal element, we are well aware, plasmid is there, transposons which are also known as jumping genes, and mobile genetic elements, and integrons, right? So these all are the genetic material which are usually having genes for drug resistance. Okay, most of the time genes for drug resistance are present on mobile genetic elements, means which can easily exchange from one bacteria to another bacteria, or which can easily be exchanged by different type of bacteria, or which can easily be moved from one bacteria to the another bacteria. Okay, but you should also know that spontaneous mutations in bacterial chromosomes sometimes also help the bacteria in order to make it drug resistant. Okay, so I hope the basics of drug resistance are clear to you from this slide. And now we are going towards our today's topic, that is mechanism of drug resistance. Okay, before going in detail about mechanisms or the strategies which are used by pathogenic bacteria to acquire drug resistance, you should be clear about some points. Okay, so on those points I have included on this slide. First point is, no particular type of resistance mechanism for a single class of drug is available. If you are thinking that any type of bacteria is going to develop resistance against penicillin, then every time it will be following this particular procedure only. Then remember, it is not like that. There is no any hard and fast rule for this, that every time if bacteria uh, is acquiring resistance against tetracyclines or streptomycin or chloramphenicol or any type of drug, there is a fixed mechanism to develop resistance against that particular drug. Okay, it is not like that. Second point, two bacteria may use different mechanism against same drug. If you are thinking that a drug is common, penicillin, so for penicillin, every time the bacteria will be using this particular mechanism. If bacteria is going to develop resistance against sulfonamide, every time each type of bacteria, irrespective of the type of bacteria, they will be following the same kind of resistance mechanism. So it is not fixed. Two bacteria may use different resistance mechanisms against same drug. Okay, for same drug, bacteria can use different type of resistance mechanism. And third point you should know, sometimes what happen when bacteria come in contact with a particular drug, during that time resistant mutants can arise spontaneously. Okay, mutation can be there in the genetic material of bacteria, as I have discussed on previous slide, in bacterial chromosome, and those resistant mutants will arise spontaneously in the presence of drug, and then they will be selected. Okay, so this is another point you should be updated with. And now we are going towards the strategies. Okay, mechanisms, strategies. So now we are coming toward these strategies which are used for drug resistance by different pathogens. So remember, strategies are of different types. And now what are these different types of strategies? One by one we are going to cover these strategies. So first strategy is what? Preventing entrance of the drug. Okay, so here what, as its name is indicating, bacteria or the pathogenic bacteria, here I can say, it will prevent the entry of the drug. It will not allow the drug to enter inside the cell. Okay. Second strategy which is used by pathogenic bacteria is to pump the drug out of the cell after it has entered. Here, drug has already entered inside the bacterial cell, but bacteria will pump it out. Okay, so that drug should not show any negative response on the bacterial cell. Now, what is the third strategy? Third strategy, inactivating or altering the drugs. Okay, bacteria can alter or inactivate the drugs. Okay, and fourth strategy is modifying the target. Of course, there is a target on which antimicrobial drug is going to act. So what here pathogenic microorganism will be doing? It will be modifying the target. Now all these strategies I will tell you one by one with the help of a diagram. Okay, so let's see, here I have drawn a bacterial cell. Okay, now we are going to talk about strategy number one. So what was the first strategy? Preventing entrance of the drug. So first of all, make it clear, preventing entrance of the drug means drug is present outside the cell and bacteria will possess some kind of characteristics, some kind of properties that will not allow drug to enter inside the cell. Okay, so you see here, these blue in color, these are antibiotics. Antibiotics are present outside. Okay, and here what will be there? In bacterial cells, outer envelopes, some kind of special type of characteristic nature it will be having, some properties it will be having, which will be helping this bacterial cell to become resistant towards this antibiotic. Okay, so this outer envelope of bacteria, what it will be doing? It will not be allowing this antibiotic to enter inside the cell. It will stop the penetration of this antibiotic inside the cell, and in this way it will be preventing what? The entrance of the drug, and it will be showing resistance to this antibiotic or this drug. Okay, so this is the first strategy, first mechanism I can say, which is used by pathogenic bacteria while they are acquiring the drug resistance. So this is one means. Now we are going towards second strategy. Second strategy, here I will show you again with the help of this diagram, to pump the drug out of the cell after it has entered. Okay, so you see here I have drawn these triangles which are indicating what? Antibiotics. Antibiotics are present where? Outside, just like this. Now here in this case, what is happening? This antibiotic has gained its entry inside the cell. Okay, these antibiotics have successfully found their way to enter inside the bacterial cell. Now what is happening? We all know about the extra chromosomal genetic element present inside the bacterial cell, that is plasmid. On plasmid, what are present? Antibiotic resistance genes are there. Okay, so you see here I have shown this gene. Okay, this gene will code for what? It will code for membrane translocases. Okay, these membrane translocases, they will form a kind of efflux pump. So what this efflux pump will do? it will pump the drug out of the cell. Okay, you can see here the antibiotics which have entered initially through this efflux pump, which was actually coded, which was formed by membrane translocases coded by a plasmid coded gene. Okay, it will help in pumping out the antibiotic, okay, out of the bacterial cell. And you should know that efflux pump, what it can do here, by pumping the antibiotic out of the cell, it can completely pump out all the antibiotic, or it can also lower down the concentration of antibiotic which was actually required to kill the bacteria. Okay, it can help in lowering down the concentration of antibiotic. So this is about second strategy. Okay, now we are going towards third strategy. Third strategy is what? Inactivating or altering the drugs. Here, this type of pathogenic bacteria, if they are not having ability to prevent the entry of the drug, to pump the drug out of the cell, then they are having other option also. What is that option? Inactivating or altering the drugs. So how such kind of bacteria which use this strategy perform this task? So let's see. Again, here I have shown red in color antibiotic resistance genes. This gene will be going to code for what? It is going to code for an enzyme. This enzyme is of which nature, type of nature? It is extracellular. Here you can see it is now present outside, and now this enzyme is having antibiotic degrading properties. And now of course antibiotics are present outside the cell. When it will come in contact with this enzyme being a biological catalyst will carry out degradation of this antibiotic in order to make it non-functional. Now it is not responding, it is not going to pose any negative effect, it is not going to exert any negative effect on this cell. And in this way, by using this strategy, bacteria can become resistant towards this type of drug. Okay, now you see before moving to fourth strategy, I have included one more example of strategy number 3. So here you can see another gene is there. In this case, what will happen? Of course, here we have seen an enzyme which was going to degrade the antibiotic, and another type of enzymes are also coded by sometimes plasmids. And these enzymes, what they will be doing here, suppose antibiotic is there and it has successfully found its way in to enter inside the cell. And now this enzyme, what it will be doing, it will be modifying the structure of, or altering the structure of this antibiotic by incorporating some functional groups, by chemically modifying its structure, by acetylation, by adenylation. So that I will be discussing in upcoming slides. So this is what, this is the, these two are the type of examples coming under inactivating or altering drugs. Okay, strategy. Now we are going towards number 4, that is modifying target. Of course, antibiotic is there, it is having some target. Okay, suppose here I am going to show you diagrammatically strategy number 4, how bacteria use it. So here you can see, suppose this is the target, which actually it is the target site for antibiotic. Any antibiotic is present outside, it needs to bind to this particular target site, only then it can make the bacteria inactive. So what such kind of bacteria which are going to use fourth number strategy, they will be doing, they will be modifying their targets. Okay, target modification will lead to what? You can see this is altered actually antibiotic target. Here antibiotics are there, they are going to, they have entered inside the bacterial cell. Of course desirable target site they are not getting because it is now modified, and it will come outside because target site is not there. Okay, which was required for bacteria, which was required by antibiotic to make the bacteria inactive. Okay, so just here I can say this is just an overview of different strategies what I have tried to cover here through this diagram. Let's take some examples now, each of these strategies. Okay, if we are saying preventing entrance of the drug, then which antibiotics are there, what kind of envelope changes bacteria actually made to acquire such kind of resistance. If we are saying efflux pump, so what is the nature of efflux pump actually, that we need to know about it. Inactivating or altering drugs, if we are saying what kind of antibiotics are there against which bacteria actually develops resistance using this strategy, how target is modified. Okay, so with the help of some examples, now we can better understand or we can better update our knowledge regarding these strategies. So let's take each of these strategy now one by one with some examples. Okay. So first of all, preventing entrance of the drug. Okay, here I have also pasted a picture so that you can recall the thing about this point, this strategy we are going to talk here. Okay, so preventing entrance of the drug. One very good example is what? Presence of envelopes outer membrane. We all are aware about this, that gram-negative bacteria are having outer membrane. Okay, present outside there, present as a part of their cellular envelope. Okay, so you should know the presence of this outer membrane actually inhibit the penetration of penicillin G in many gram-negative bacteria. That is why that many gram-negative bacteria, they are resistant to penicillin G, because this outer membrane is actually acting as a barrier. It is not going to provide penicillin G the particular binding site so that it can enter inside the bacterial cell and make them inactive. So this is the first point you should know. Second point, changes in penicillin binding protein. Some bacteria, what they do, they make changes in penicillin binding proteins. In penicillin, we all are aware, if this antibiotic needs to act on the bacterial cell to make it inactive, it needs to bind on some specific proteins. And bacteria, what they generally do, some bacteria carry out genetic mutation in these proteins, and it will inhibit the binding of penicillin to such proteins. So bacteria will be no more sensitive towards this antibiotic, and in this way it will develop resistance. Now you see third point, decrease in permeability. Okay, outer envelope permeability is very commonly used by many bacteria which develop sulfonamide resistance. Okay, now you see fourth point, high content of mycolic acid in a complex lipid layer outside the peptidoglycan. So you all are knowing, I think many of you are knowing that mycolic acid is present in the cellular envelope of mycobacterium, and you should know that the presence of mycolic acid make the mycobacterium impermeable to various water soluble drugs, and in this way it help in um, uh, development of resistance in mycobacteria, right? So you see, I hope with the help of these examples this strategy is very well clear to you now, that how this strategy is used by bacteria. Okay, in terms of outer membrane, by making changes in penicillin binding protein, by changing permeability, and by presence of some special type of components like mycolic acid, right? So now you see we are going towards second strategy, to pump the drug out of the cell after it has been entered, right? So here again I have pasted this picture so that you can better recall this point we have already discussed. Now we are talking about the efflux pump and all that, right? So you see, sorry, now you see efflux pumps. What are actually these efflux pumps? Some pathogens have plasma membrane translocases that expel drugs. So with the help of this diagram, initially we are clear about it, that membrane translocases which usually act as efflux pump, they are coded by antibiotic resistance genes which are generally present on mobile genetic elements. Okay, so these membrane translocases are called as efflux pumps, and most of the times what is there, these efflux pumps, these membrane translocases, they are actually non-specific. Okay, they can pump many type of drugs out of the cell. Okay, and making the bacteria multi-drug resistance. Okay, multiple drug resistance is a very common issue. So if such kind of efflux pumps are non-specific, means they are not specific for a particular drug, then they can offer advantage, to bacteria, to pathogenic bacteria, to pump multiple type of drugs, and in this way it will help in developing multiple drug resistance, means same bacteria can show resistance towards different drugs. But remember, presence of non-specific efflux pumps is very much common, but still selective pumps are also there in some bacteria. Okay, next point, you should know drug proton antiporters. What actually it is, as its name is indicating here, what will happen if drug has gained its entry inside the cell, it will be pumped outside, it will go outside if proton will come inside the cell. Okay, so these are known as antiporters, means one is coming inside, then other is finding its way outside. So protons enter the cell as the drug leaves. Such type of antiporters have been reported in E. coli, Pseudomonas aeruginosa and Staphylococcus aureus for development of drug resistance. So I hope this point is clear to you now that to pump the drug out of the cell after it has entered, how bacteria use this strategy? It can be used in the form of efflux pumps. Efflux pumps can be selective or they can be non-specific, or in the form of antipotents, right? So now we are coming towards third strategy, that is inactivating or altering drugs. So again, you are already clear about this picture, right? Which we have discussed. So now we are talking about these points. So you see inactivating, altering drugs. So first point you should know here, I have discussed about gene which are coding for some enzyme. Enzymes are having antibiotic degrading properties. So one such enzyme is penicillinase. Penicillinase carry out what? It carry out hydrolysis of beta lactam ring of penicillins. Beta lactam group of antibiotics are there, and penicillin is one of the very good example. So it will carry out hydrolysis of this ring, right? Another example, chloramphenicol acyl transferase. So what it will be doing? You should know that chloramphenicol is an antibiotic which contains two hydroxyl groups. So these hydroxyl group, they will be actually modified. We can say this is a type of chemical modification, right, or functional group modification, which is carried out by bacteria to acquire resistance. Okay, so it means alteration of drug is there. Chloramphenicol is having two hydroxyl group. They can be acetylated in a catalyzed reaction with the help of acyl transferase enzyme, right? Sorry, this will be acylated. Okay. Now you see third example, aminoglycosides acetyl transferase. Here what will be there? Again, acetyl transferase enzyme is there, which will be transferring acetyl group to aminoglycosides. Okay, aminoglycoside represents a group of antibiotic which include kanamycin, gentamicin. The bacteria which actually develop resistance against aminoglycosides, they use such kind of enzymes. How they use such kind of enzyme? They use such kind of enzymes to carry out acetylation of amino group of aminoglycosides. Okay, so now you see next example, phosphotransferases and adenyl transferases. These represents other type of enzymes which are expressed by various type of bacteria to develop resistance against, again, aminoglycosides. Aminoglycosides are also having what? They are also having hydroxyl groups, and to these hydroxyl groups, phosphotransferases can carry out, can incorporate, sorry, phosphate group, and adenyl transferases, they can incorporate adenyl groups, and in this way they will change the structure of the antibiotic in order to make the antibiotic inactive, and in this way microorganism will be developing resistance. I hope with the help of these examples you are clear. So here I can say what are the mechanisms mainly I have discussed. Degradation of antibiotics, so penicillinase is the example of this, or chemical modification of the antibiotic or by incorporating some special type of functional groups or some special type of chemical moieties on the natural structure of antibiotics, which is making them um, inactive. Okay, so these enzymes are also expressed by bacteria to acquire resistance against different drugs. So now you see fourth strategy, that is modifying target. Okay, so modifying target, here we have discussed about this, so here I am going to include some examples. So you see changing the affinity of ribosomes for erythromycin and chloramphenicol. So many of you I think are knowing about it, that erythromycin and chloramphenicol, they are actually protein synthesis inhibitors. Okay, they inhibit the process of protein synthesis after binding to 50s subunit of 70s ribosome of bacteria. Okay, and in 50s there is 23s rRNA sequence. Okay, so what will be there? They are actually specific to 23s rRNA, which is a part of 50s subunit of bacterial ribosome. So how bacteria respond towards such kind of drug? If they are showing resistant toward these drugs, bacteria actually make changes in their 23s rRNA sequence, which is a specific or which is highly required for erythromycin and chloramphenicol to act. Okay, so if bacteria will make changes in this kind of sequences, then antibiotics cannot successfully bind to 23s rRNA, and they will not be able to exert their effect of protein synthesis inhibition, and bacteria will become resistant toward this drug. Now you see second example, changing the terminal D-alanine in their peptidoglycan to a D-alanine D-lactate. Which bacteria use this type of change? So we first make it clear, we all are clear about this point, that peptidoglycan, which actually form bacterial cell wall, it is made up of two sugars, complex sugars NAG and NAM, N-acetyl glucosamine and N-acetyl muramic acid. So I hope you all are clear about it, that NAM unit, out of NAG and NAM, on NAM pentapeptide chain is there, right? Pentapeptide chain which is having 5 amino acids, and its terminal amino acid of this chain include D-alanine, and now it will be changed to D-alanine and D-lactate by which bacteria they will be changed from this to this sequence? By enterococci. Why enterococci make this change in terminal sequence of pentapeptide chain attached to the NAM? It will be helping the enterococci to show resistance towards vancomycin, because vancomycin actually has more affinity for binding to D-alanine and D-alanine. And when this sequence is changed, the order of the type of amino acid is changed. Of course, its binding they will have less affinity for binding with the vancomycin, and enterococci will become resistant towards this particular drug. Okay, so now you see third example, alteration of susceptible enzymes. Okay, so what will be there? Sulfonamide actually act on the enzyme which is involved in folic acid synthesis, and the bacteria which develop resistance against sulfonamide resistance, they actually express such type of enzyme for synthesis of folic acid which has lower affinity for sulfonamides. Okay, so in this way bacteria will develop resistant towards sulfonamide. So now take next example, alternate pathways. What does it mean? Suppose a particular type of antibiotic is there, and it is going to act on a particular sequence or a particular step in metabolic pathway. Then what bacteria can do? Bacteria can bypass the sequence inhibited by the agent. Okay, so one example we are already clear, sulfonamide. Sulfonamide act on mainly target the folic acid synthesis process. So bacteria, if they are acquiring resistance against sulfonamide, what generally they do? They stop the synthesis of folic acid. They stop following the pathway of folic acid synthesis. What they do? They start to fulfill their requirement of folic acid from the surroundings. Okay, so you see some bacteria are resistant to sulfonamide simply because they use the preformed folic acid from their surrounding rather than synthesizing it themselves. So they actually bypass the sequence inhibited by the antimicrobial agent. Another example, increase in production of target metabolite. So another type of option is what, which is actually availed by bacteria, that some strains of bacteria, they actually develop the resistance by increasing the rate of folic acid production. In this way, what they will be doing? They will be just in this way counteracting the sulfonamide inhibition. So I hope all four strategies are clear to you all. Thank you so much. Keep watching and stay connected.